So our next speaker is Rodgers from Oxford, and he's going to talk about the quantum mechanics of bird navigation.
Slightly provocative title.
Yeah.
Okay, so I am a physical chemist by training, and so I wanted to sort of give you a physical chemist's perspective on this. I wanted to convince you from experiments done in the lab that these kinds of magnetic field effects actually happen, and that they're in non-biological systems, are very reproducible, and they actually tell us an awful lot about chemistry in systems and have been very useful. Torsten's already gone over this really, but I want to just come back to this radical pair mechanism and talk about maybe from the chemistry experiments in the lab what actually are the key things as far as we can tell at the moment if you actually want to set up a reaction which will show a strong magnetic field effect, and then kind of by extension if you wanted to show a compass effect, what do you have to do? And then also just talk a little bit about sort of our take on the biology. So there are actually lots of magnetic field effects in the chemistry world. This is not the kind of magnetic field effect that I'm going to talk about, but I just wanted to show you because I think it's often something people don't really believe. So if you take a Petri dish which is filled with cobalt EDTA, this very pale pink solution, and you drip hydrogen peroxide into it, this sets up a catalytic reaction that oxidizes the cobalt-2 into blue cobalt-3. And what's quite amazing about this is that if you drop the drip into the middle of the dish, you can see this circular region that expands. It's very, very obvious. It's a blue blob that expands until it fills the whole of the Petri dish. It takes a couple of minutes. If you put a strong horseshoe magnet underneath the Petri dish and drop the blob in the middle, then actually this thing expands out, it avoids the poles of the magnet, it flows all the way around the edge of the Petri dish until finally it comes into the regions of strong magnetic field. So it's kind of, there are a lot of magnetic effects in chemistry, even though you might not expect there to be too. And they all kind of involve some sort of kinetic effect. So in this case, these things are chemical waves, and the reason why it works is because it's autocatalytic. So you have very small disruptions in the way in which that chemical wavefront propagates, and it gets amplified as the thing goes on. Okay, if you want to go to still weaker magnetic fields, then there really is only one mechanism that we know that causes these kinds of effects. And so this is what I'm going to talk about for the next few minutes. So as Torsten said, that the basic mechanism is that you have some sort of precursor, and you have to give it a significant amount of energy when you excite it. And this is important precisely because of this idea of getting out of the thermal limit. So we kick the system a lot. It's often done with light, or in the 70s there was a big body of research looking at pulse radiolysis. So you zap X-rays into something and you then watch what happens in the radiation spurs. Sometimes you can also get away with heating things to make some sort of proton transfer or something like that happen. And the key thing here is that often these excitation steps, so for example, if you do electron transfer after you've shone light into a system, actually that process, that chemical reaction, conserves the electron spin. And so since, in most of the times, the precursor that you start with is a ground state closed shell molecule, then all the electron spins were paired up in that molecule. And so when you hop one electron over to somewhere else, it has to be in a singlet state, even though these two things are now on different molecules. And then what happens over the following, usually nanoseconds, perhaps microseconds, is that there's an evolution of these. So usually there are some decay pathways, and the key thing is that sometimes you can have something different happen to the singlet state versus the triplet state. The most obvious different thing you can have is the back electron transfer, because if you flip the electron spin over, then it's spin forbidden to put the two electrons back into the same orbital to make the precursor. And so typically what you find is that at the very least you see a lifetime effect as we decay back to the precursor that we started with. And then in more interesting systems, if you can rig it carefully enough so that something else can happen, maybe you react with the solvent and you go off to do something else, or, and then the possibilities are there. So in the simplest system, if we just imagine a radical which basically has one hydrogen atom on one of the radicals that has a hyperfine coupling, this sort of illustrates to you what's actually happening in detail in the system to get a magnetic field effect. And so what you can see is that In the system completely on its own, with just the electrons, the two unpaired electrons, you see this beating back and forwards, the green line. And this is the interaction of the hyperfine coupling and the Zeeman, the hyperfine coupling with the spin states. And this drives it, oscillates backwards and forwards, and the frequency is the hyperfine coupling. If there's some chemistry that goes on as well, then obviously this singlet state is decaying and it's being consumed, so that would be the red curve. And if we integrate under the red curve, we see a yield that builds up until some final time. Now if we turn on a weak magnetic field, then the probability of being in the triplet state in this case, we get a beating effect. So it beats backwards and forwards because of the difference between the hyperfine coupling and the Zeeman interaction that you've applied. And that can have a very significant effect if it's at the right sort of frequency to significantly alter this early part of the dynamics while the decay dies off very quickly. And so you can begin to see that there are going to be restrictions. If it takes forever for this thing to decay, it's a very slow reaction, then actually on average over a long period of lots and lots of cycles, the average of this green beat pattern looks just the same as the pattern up here, and you see no field effect. And conversely, if you have very, very, very fast dynamics, you don't have long enough to build up this pattern of beating. Okay, so in a radical pair, what things do we put in? And this is a fragment from a molecule that I'll talk about a bit later. And it's kind of all the things that we would expect from EPR NMR. We see the Zeeman interaction, so that's the electrons with the fields. The nuclei also would interact with the external magnetic field, but of course that interaction is so weak that it really hasn't got anything to do with this. The hyperfine interactions, both the isotropic part and also the anisotropic part, which depends on the shape of the molecular orbitals in the molecule, and that will be key when we talk about the compass. And then other interactions, the electron pair has a dipolar interaction, the magnetic interaction through space, and also an exchange interaction, which is through orbital overlap, and that's quite difficult to model actually in these solution phase things. It's really, very difficult to calculate, and because it varies so rapidly with distance. And so we tend to either ignore this or use a very simple phenomenological form. And then in some systems with heavy atoms, spin-orbit coupling as well, and there's a family of magnetic field effects where spin-orbit coupling is the key thing that drives it. Okay, so I think I've already kind of got ahead of myself and mentioned this, but basically we have to set the kinetics up properly. There has to be a decent competition between the reaction that's going to go off and form the product from the singlet state and the triplet state, typically maybe a factor of 10 times in the rate constants at most. Otherwise the reaction will go all one way effectively, and it's pretty much irreversible. And it's got to not take so long that either relaxation dominates and basically you see no effect, or so that there's not enough time, or so that the long time average of the beats means that there's nothing really going on. Okay, so if I've got to have a radical pair, then I can also perhaps try and say something about the geometry of the system. And so it's quite interesting actually that both the exchange interaction and dipolar interactions are very strong if the electrons are close to each other. And they act to, they change the eigenstates of the system, and they quench this singlet triplet interconversion. And so what's quite interesting actually is that there's a bit of a sort of critical region really. And so what you can actually say is that there's a kind of a minimum distance under which the exchange interaction is so strong that you get no singlet triplet interconversion. But then equally, you can look at the idea of actually saying, well, if I want to move the things really far apart, then you know from your work in the photosynthetic reaction centre that the rate of that electron transfer will drop down to nothing. And I said that the kinetics had to be very finely balanced, and so the magnetic field effect would disappear if the two radical centres was too far apart as well. And we find with a sort of back of the envelope calculation that actually what we want is a distance that actually corresponds quite nicely to what we see in some of these flavour proteins like cryptochromes. Another thing which is obviously very important if you spend more than a moment thinking about this is, well, do these radicals move? Because, as Torsten said, if we want a compass, it's not going to be much good as a compass if the radicals are freely moving around. I mean, imagine a ship's compass if the dial is just not attached properly and it's free to roll around. You won't be able to tell anything. And so in the case of the reactions, actually you can be a bit more specific about that, and you can look at it with, well, it's not quite Redfield theory, but you have to change the approximation slightly to account for the fact that this is running at low fields where the electron spins are not really well quantized along the external magnetic field like they would be in a higher field EPR or NMR experiment. And what we can see is that actually, okay, what do we need? Well, we either need rotational motion that's very slow or very fast. And this extreme narrowing limit, which is the kind of thing that we typically work out for NMR experiments, that's so fast here that actually it's not really reasonable to think of things rotating on 100 picosecond scale. The vibrations, of course, happen very quickly like that and aren't a problem. Don't cause very rapid relaxation of the coherence in the radical pairs. And being slower than 1 microsecond, I guess in a biological system that's kind of beginning to mean fixed. So we've got this picture of needing the radicals to be kind of pinned into some matrix, a protein fixed in a membrane, fixed in relation to the animal. It's also true that you can imagine, if you do the maths, that maybe you could be lucky, and maybe you could set a system up so that relaxation can actually preferentially make some of the spin states of the radical pair relax faster than others. And these kinds of effects are observed, it's called the Triplet Mechanism, and you see these in some solid state physics systems, and they actually are the source of magnetic field effects. But that's quite a specialised thing, and we've done quite a few calculations in systems that would be more suitable for the bird compass, and it really seems that no, we're not in a regime where relaxation helps. Relaxation always seems to squash away the directionality that we observe. And this is the kind of thing I meant there. Okay, so that's the recipe. Does it work in practice? What sort of systems does it work in practice? The earliest experiments were done in doped anthracene crystals, where you can get charge separation. Magnetic field effects in modified photosynthetic reaction centres, where you block the forward electron transfer in order to adjust the kinetics so that you get a good competition, as I was talking about. And in liquids, there's a whole family of radical ion pairs, so things where the electron transfers happen, which means there are charges, plus and minus, on the two radicals, like I drew. And that's fantastic because it constrains diffusion. It means that there's a strong long-range Coulombic interaction which encourages the radicals not to part forever, but to move apart for a bit and then to come back, and then we can see whether they'll recombine or go their separate ways. If we look at the mechanisms of these radical pair effects, we can see that at very low fields, so something on the order of a millitesla, this is sort of where the Earth's magnetic field is, right down at the bottom corner of this graph, we often find that singlet products decrease in yield slightly. Then the yield tends to increase until we get this long region over plateau. And then high field experiments done in NMR magnet, big superconducting magnets, they tend to see a different mechanism due to the G factors differing on the two radicals. And those field effects generally see a reduction in the singlet yield. Okay, so if we wanted to look at a field effect that happens on the bench in the lab, it's the same scheme I told you before, and just sort of drawn around slightly differently. And so the key thing here is that for liquids, we form this radical pair, they're in close proximity to each other, we shine in ultraviolet light to make the electron transfer happen. Only the singlet radical ion pair can go back and fluoresce. The triplet radical pair can't, and so it's effectively stuck, and the radicals will just drift apart and react to form some background product with the solvent. And then the key thing is that as the radicals start to diffuse apart, they reach a region where the exchange interaction is no longer very strong, and they can actually start to evolve and do some interesting spin dynamics. And then if they move back into close proximity again, that singlet triplet interconversion is frozen out because the exchange interaction becomes strong. And so you can think of the sort of simplest mathematical model. I mean, it's very basic, and it seems that it should be too simple, but actually this is good enough to model things. And just a density matrix, which we start off in a singlet state for the electrons, and in a completely uncorrelated state for all the nuclear spins. And then we just follow that to follow and work out what the probability of still being in singlet state after some time t is. and then we combine that with some model of the kinetics. And actually, again, it's surprising, but you can put in sort of reasonably realistic models of what would happen for diffusive motion in these charged systems. And actually, this simple model, first order kinetics, actually often beats them hands down. And that's perhaps still an open question why that happens. And then you can tweak lots of parameters about the system. So to tweak the kinetics, you can change the temperature, and you can change how viscous the solvent is, and all sorts of things like that to come up with a picture. And then you can do a variety of experiments that, for historical reasons, all seem to have their own acronym. But basically, you can vary the strength of a static field and see how does the reaction yield change. You can put radio frequency fields there. You can vary the frequency of that field. You can change the strength of the field. You can adjust what you put in the bottle, and any combination of the above. So here's a typical bit of apparatus from the bench, and this experiment looks at an aromatic molecule, pyrene, it's quite carcinogenic, and dicyanobenzene. And these systems are nice because they have very long-lived exciplex state, which then fluoresces. And so what you do is you shine in ultraviolet light, you have the sample of stuff flowing through so that it doesn't get photobleached, and you look at the fluorescence coming out in a perpendicular direction. And what do you see? Well, this is an example of a spectrum where you've varied the static field from 1 up to 4 millitesla in the presence of a radio frequency field. And what you see is effectively a sort of way of measuring the EPR spectrum of both of the radicals at once. And the take-home thing about this is really kind of picked out at random. It's just to show you that even that kind of simple-minded way of calculating them, it runs very quickly, and it gives really very good qualitative agreement, at least, with the shape of the spectrum. And you can go on, so this is a related system, so we've deuterated the pyrene, and we've chosen one particular isomer of the dicyanobenzene. And you can see here that this is the experimental signal in one particular case, and this is showing the difference between using linearly polarized radio frequency field and left and right hand circularly polarized fields. And then just to show you how sensitive the simulations are really, if I adjust the strength and the simulations of the radio frequency field, we can really see, well, this one here actually has the right kind of line shape. And it gives you a sense of the fact that it's not just that any parameters we put in make the right curve. These don't because if they have that many features, but actually they are very sensitive to the parameters that you put in. And there aren't that many free parameters in the model because we can calculate all of the hyperfine couplings trivially with quantum chemistry program, or we can measure them by EPR. There are other effects, so you can look at the difference, see what happens when you deuterate things, and that's quite nice because you wouldn't expect it to change the kinetics, but you obviously expect, since the magnetogyric ratio of hydrogen and deuterium differs by about 6.5 times, this is quite a nice way of changing the hyperfine coupling constants in a system where you can be very confident that the kinetics have stayed the same. And again, the take-home message here, the dots are experiment, and the line that goes directly through them is theory. And in this system, it's just looking at the effect for static fields, and that's a bit easier to calculate. And so really, you can get things down very nicely. And this is a blow-up showing what happens in the region of very weak magnetic fields, sort of less than about 2 millitesla. Okay, so let's move a little bit towards biology, since that's what I was supposed to be telling you about. We know about magnetic field effects in modified photosynthetic reaction centres. You have to be a little bit careful of this because you always come across a sort of steady stream of effects, magnetic bracelets that cure you from cancer and all these kinds of things. So there's a huge amount of bunk out there, but there is also a huge amount of stuff which is robust and is scientific. There are a number of reports of magnetic field effects on enzyme reactions, and as Torsten's been telling you, effects on the compass sense of birds, in particular this guy, the European robin. So I think I won't dwell on this. This was just the key finding from the Wiltschko's back in Science back all those years ago in about 1970. And this was the idea that if the bird is heading this way in the normal Earth's magnetic field, if I swap the direction completely, the bird flies in the same way. But if you change the dip angle, that's what makes it go backwards, as Torsten said. And so the key finding of that, remember, was that we sensed the axis of the magnetic field, or the birds did, but not which way was north and which way was south. And then a lot of other ingenious experiments. The success of birds depends on the colour of the light. You have to not cover their eyes. You can only have a reasonably narrow range of magnetic fields over which they can actually perform this feat. So if you put them in a magnetic field 10 times stronger than the Earth's magnetic field, that confuses them. They can't work out which way is north anymore. And so we think that the evidence is accumulating that this comes down to this idea of having the same chemistry that we saw in the lab running in a protein, perhaps cryptochrome, embedded somewhere in the retina, and Torsten's model of how this might turn over into a visual pattern. So if you plot the anisotropy of the reaction yield for a particular radical pair, so basically you pick a direction and put the magnetic field along that direction. If it's red, it means that there's more singlet product formed than average, and if it's blue, it means there's less. And if you imagine that process was a step in vision, then it's a bit of a sort of a cartoon picture maybe, but you could actually imagine, okay, well maybe this is what the bird kind of sees as a result of the magnetic field. And then in terms of disorder, we often find very complicated patterns. So this is a radical pair taken with a flavin molecule and an amino acid, so something like there is in cryptochrome, and you can see a pattern that really looks completely unintelligible. But then this plot C and this plot D are actually the same figure. It's just that in this one I've rotationally averaged it around the z-axis. And so in fact in some senses it might actually be that not having the molecules completely pinned in place might actually help us. Because if you had disorder in a couple of the directions but still had a fixed axis, you could actually make the patterns that the bird would see simpler and more easy to understand how they might be interpreted. Actually doing calculations on this doesn't scale very well, and I've been interested to hear some people talking about that kind of problem, because of course as we run this you start wanting to put in all these anisotropic hyperfine couplings. They tend not to be equivalent to each other, any of them, and also you sometimes have other things like nitrogen nitrogen atoms in there which are spin 1, and so you can very quickly accumulate enormous density matrices and it quickly becomes impossible. And so we have to be quite sort of judicious about the calculations we do. And it's amazing in a way, really, how much you can still get from the simplest calculations where you just put one proton on one of the radicals and leave the electron on its own on the other. And to give you an example of some of the things that you can measure, if you measure the kind of the anisotropies, this is some metric of anisotropy, as a function of magnetic field strength, you can see that you can get very sharp resonant responses, and that kind of seems like what the birds do, the fact that they can only orient properly over narrow ranges of field strength. And you can look and you can see that, well, the pattern is complicated. And Torsten mentioned that actually these anisotropic responses can be quite immune to adding isotropic hyperfine couplings. And the reason is, it's because the anisotropic responses just very slightly perturb the way these levels pan out, and as they go from the zero field states up towards the sort of intermediate field states, you get this little forest of level crossings that cause that first dip in the magnetic field effect curve. And these are critically sensitive to the orientation in a system which is anisotropic, but they don't actually depend that much on the isotropic parts of the hyperfine interaction. You can show frequency response as well, and I think that it's really nice in terms of the experiments that Torsten was talking about here, of actually kind of doing in vivo EPR almost on a bird and trying to see what's going on with the radicals actually in biology. And I'm very impressed that this experiment works at all. It's such a difficult thing to do. Okay, so far I've shown you that we can see magnetic field effects in the lab. I've shown you that there's a body of theory that explains them. And that actually we can be, reasonably good at quantifying what we see. But it would still be nice to see something at the Earth's magnetic field, because that really does seem, it's quite hard to believe, isn't it, that such a weak interaction could actually produce a measurable effect on chemistry. And so if you want to see something like that, well, you need to go back to the recipe I was talking about at the start, and you pick your way through and try and find a molecule which is going to have a very long lifetime, so that you've got a long time for this singlet triplet interconversion to happen. And this molecule is something that seems like a very suitable thing. So if you shine light on this, you get an electron transfers from this carotenoid here over onto the fullerene. And this molecule is also quite nice because fullerene, of course, has no hydrogen atoms there. And since carbon-13 is only 1% natural abundance, effectively this electron over here really is pretty much on its own. It's just existing. And then this half of the molecule has all of these conjugated double bonds. And that means that the carotenoid is planar, and that has very nice properties in terms of the anisotropy, because it means that you get this pi electron cloud above and below the molecule, and you get very strong, very well-directed anisotropy in this part of the molecule, and then essentially nothing over here. So you can almost think of this molecule as a sort of reference and a probe kind of thing. So in this side of the molecule, the strongest interaction is with the external field, and that's kind of a probe. And in this side of the molecule, it's got strong interactions internally, and so it goes about its business, quite happily itself. So chemically, to actually look at this on the bench, this shows the apparatus, and it's a pump probe experiment. So you pump in with a pulsed laser to drive that electron transfer, and then you probe it, looking at the UV absorption of one of the excited states. And actually, the photochemistry of that triad molecule is very well understood. And then you can measure it with field and without field, and then subtract to see what effect the magnetic field had. So this first figure shows us if we just dial the magnetic field to 0 microtesla. So this is a control experiment, and it's reassuring to see that as the time goes after the laser pulse, really there's nothing significant going on here. Whereas if we apply a field of 39 microtesla, actually we're beginning to see some dynamics. And by 49 microtesla, and this is kind of the Earth's magnetic field strength in Oxford, really you can begin to see quite significant effects on the downstream photochemistry of this triad. And I just find it amazing because a little magnet like this that you could put on the desk has 20,000 times stronger magnetic field than the Earth's field, which is causing these chemical changes. Okay, so that's kind of a chemical reaction at least, which is sensitive to an Earth-strength magnetic field, but it's still not a compass. Is it actually possible to get compass information out of a system that isn't a kind of a solid-state crystal of something? And the answer is yes. So what you need to do for the triad molecules is you freeze them down so that they're relatively immobile to liquid nitrogen temperatures, and then you can do photoselection. So if you polarize the probe laser that comes through, probe at a different frequency this time, but we're still monitoring just a different part of the absorption spectrum of the excited state. And then what we pick out is only molecules where the transition dipole is in the right direction. And we can change the polarization here, or more easily, we can just sweep by using a pair of Helmholtz coils, these two here, the inner ones and the outer ones. We can sweep around the direction of the magnetic field. And so what you see is, again, some control experiments showing no effect, but actually you can see this orientation dependence. And if you plot that out as a polar plot, it really looks quite like the polar plots, even from these most simple systems with just a few hyperfine couplings that you begin to drop in. And that's quite exciting. It's not an Earth-strength magnetic field, you have to crank it up a little bit more. But at least there you've kind of got the ingredients. You've got some chemistry that works at the Earth's magnetic field strength, and something that is at least capable of forming a compass. So coming back more to the biology, I think that these EPR experiments are actually quite significant in a way, because it's very difficult to prove that you have radical pairs, and even more, it's difficult to prove that this spin correlation that keeps it really in a singlet state, in a triplet state, and not just in some relaxed mixture, has persisted. And magnetic field effects are a very convincing way of showing that, but another convincing way of showing that is pulsed EPR that Tustin talked about. And so in some of these pulsed EPR experiments, in ESEM, electron spin envelope echomodulation experiments, you basically excite with a laser typically, and then you apply 2 pulses spaced at a time, and then you wait to see the echo come back out in the EPR signal. And if you follow the maths through, I can give you the reference. What you see is that if you get this antiphase pattern whereas in a pair of peaks corresponding to the two electrons for the radical, if you get a situation where one of them goes up and the other one goes down, that's really very characteristic of a correlated radical pair, and that kind of behaviour is immune to a lot of the other interactions in the system that we don't really know the parameters of. It's also a way, although somehow it doesn't seem to be used so often, of measuring values for the exchange and the dipolar couplings, which are these parameters that are, well, dipolar is easy, but the exchange interaction is a difficult thing to calculate. And so what do we see? Well, photolyase is a protein very similar to cryptochrome. It's just got a different tail on the end. And it's another blue light flavor protein. And that shows these radical pairs. So this has correlated radical pairs. And you probably can't see the time scale here, but this is something on the order of about 50 microseconds. And really, I mean, that's amazing. The radical systems that I showed you earlier in the lab, the radical pairs there persist only maybe for 100 nanoseconds, something like that, and that's enough to do the chemistry. This is quite incredible. There is clearly something quite special about this family of proteins, even if from looking at them we don't understand why. And actually, a couple of years ago, the photolyases were actually shown again to have a magnetic field effect. It's a pretty subtle thing, but the difference here in the two traces between effectively zero magnetic field in the blue trace and then a slightly stronger, well, fairly strong, 39 millitesla magnetic field. And you can see the dynamics of the decay of the excited state are different in the two different magnetic fields. Cryptochromes that Torsten's talked about have this kind of structure, and people have talked about whether the precise positioning in space of three tryptophans in the protein and the flavin molecule is significant for forming these effects. It's sort of on the face of it, it seems a nice idea. You get electron hopping across this chain, and that's a neat way of getting the electrons far enough apart that they don't have this strong exchange interaction which stops the magnetic field effect, but don't have to hop in any given step so far that it's very slow. In practice, it seems a little bit as if this might not actually be the case. There was some stuff published this summer by Stephen Reppert, and he looked at systems where he'd actually been able to remove some of these tryptophans, and it didn't seem to change things that much. But it's not entirely clear. What's exciting, perhaps, again, though, is that cryptochrome does produce these time-resolved EPR spectra, and it's still got this antiphase property, which means there is a long-lived correlated radical pair. Not perhaps quite so long as in the photolyase, but still in the right kind of ballpark for showing an Earth-strength magnetic field. And then there are some biological experiments, which Torsten talked about in much more detail than me, but basically you can show that cryptochromes are present. Cryptochromes in garden warblers, which are another migratory bird, get produced more during the season when they would want to migrate, and they don't seem to get produced in close relatives of the garden warblers that are non-migratory. There have been some ideas of looking at other systems. Birds are obviously a pretty hard experimental system to work with. I think it's impressive, the level of results that they've managed to get. It would be much, much easier if you could do it on something smaller that you didn't need so much ethics for, and that you could run lots and lots of experiments the whole year round. But I guess this is just a sort of slightly sobering warning still, that it's difficult. The birds are quite a special system. The Wiltschos have built up a lot of expertise and make these experiments work well. And other people who've looked at the bird system, Henrik Moritzen in particular, have managed to reproduce quite a lot of this work. And I'm sure the bugs will be busted out of this kind of thing, but we aren't there yet with another model system that really lets us push forward and do lots of, it would be nice to do lots of molecular biology and stuff and knock the different genes out and test all these things, but not quite. So would cryptochrome really work? Well, probably, we think. But it's difficult until you know exactly what's going on. It's sort of chicken and egg situation. If you can tell me exactly how it works, then we can calculate and we'll tell you that it worked. But until we exactly know what's going on, for example, no one knows what the photocycle of cryptochrome is. It's not completely clear actually what reaction is happening. What are the different species that are involved as well as the protein? And that's really critical because we need to know their parameters in order to put them into all the simulations. But we're getting closer. And I think I will stop. Goodbye, thanks.
Thank you.
Person said it, this is a coherent quantum process, so the evidence is because you do a time resolved EPR experiment and you see that the process occurs during the dephasing time. Is that the point? Did I get that right? Why is it coherent? What is the issue about quantum coherence here?
The issue about quantum coherence is that you need to have a distinction between the singlet and the triplet state of this radical pair.
Yeah.
And that's something that they don't really differ in energy because you've removed them a long way from each other. So, right. So the claim is that this part needs to be coherent because actually these radicals can move quite a long distance apart from each other, especially in the solution phase. And so if there weren't a coherent state, you do get other systems where, for example, if you have oxygen atoms in there, oxygen has strong spin-orbit coupling. It tends to produce very rapid relaxation. And so you can have a system where very quickly after separating from each other, they forget, basically, what the relationship was between the spins on the two molecules. And it doesn't matter one blind bit while they're floating around in solution, but it matters critically as they come back together and we want to see whether the back electron transfer happens. Was it still a singlet state or had it become a triplet state? And that's the claim why it's a coherent mechanism.
In these cases where you look at the effect of weak magnetic field, is the effect you're seeing proportional to the square of the field amplitude as I would expect, or is there anything else?
It's not as simple as that. I mean, in fact, the spectra have quite a bit of detail. I mean, you saw some of the simulations that I showed. There's in some systems, there's a very short time approximation way back when from the early days of the theory, which is a bit like one of these t-squared things that I saw someone else talk about. And in that kind of situation, I think, yes, just in the very first bit of the decay, you can get simple rules that relate the field strength to the time. But that's not really interesting, because what you actually need to do is to integrate, enough of this complicated pattern of beats. And actually, all reasonable molecules have 50 hydrogens in them or something like that, between the pair. And so the pattern of beating here in the singlet probability is immensely complicated.
My colleague Max Crosley has made a large series of fullerene, quarter and ferrocene triads, tetrads, etc. that show sub-picosecond singlet charge recombination and post-millisecond triplet charge recombinations. You're interested in this sort of molecule as a model system?
Yeah, I mean, if you send me the details, I'm sure that would be of interest.
In your figure that you have the quantum yield as a function of the magnetic field, there is a region in which the reaction yield is the same for quite a while in this one. Does it mean that the coherence, the correlation between the two spins is the same along the? Does it vary? Does it depend on this magnetic field? Essentially, the question is: the quantum coherence that we're talking about here the same, independently of the field?
I think so. I mean, the reason why you get this plateau is because you've got to have an effective competition. So you've got to have a competition between the Zeeman effect coming from, the magnetic field and the kinetics of the system, and then that's kind of got to be similar energy to something else, something kind of something to refer against. And just in most organic molecules, the hyperfine couplings are not that strong, and they all tend to fall in this sort of few millitesla kind of region. And then the next biggest interaction in the system is the delta G mechanism. So basically, small spin orbit couplings which change the energy of the electron. And so I think it's more to do with that, with the details of the beating pattern, than necessarily with whether it's still coherent or not. It shouldn't change that. I mean, relaxation changes as we move across here, and relaxation gets slower as we move into stronger field region. But it's difficult to know how you would measure it, because you don't see such strong effects, really.
More questions? If not,